Hey everybody, we're going to do a face recording on this one just because it's kind of boring. There's not a whole lot of pictures for you to see, but we're going to run through your genetics introduction notes. We're going to get about, we'll go down through genotype and phenotype. Okay, so we'll get down to genotype and phenotype. We may get to gene and its relationship to alleles, but we might save that for more of a face-to-face thing. Alright, so let's look at Mendel's work. So Gregor Mendel, the father of genetics, right? So who is Gregor Mendel? He's the father of genetics, and he's the guy that figured out the rules for heredity. And heredity is how are things inherited? How did you get your genes from your parents, and so on. And he did this by doing plant breeding. So he bred plants for over 2 years before he actually started to really collect data. So he observed. He did really good scientific work by observing repeatedly for years and years and years. And as he was working, he noticed several things. The first of these is there are always two forms of a trait, and those two forms of a trait are called alleles. Now, hopefully this is a term that's familiar to you from middle school. Two forms of a trait are called alleles. So those are the two forms of a trait. And he noticed that one of the forms of the trait always showed up. He called that one the dominant one. The other one that got masked, he called the recessive trait. As I'm writing, this is shaking a little bit, but you get the idea. So the recessive trait is the one that was covered up by the dominant traits. So he figured out sometimes you compare two dominants and end up with a recessive showing up for some reason in the offspring, which are the kids from the cross. And he also noticed you didn't really get a blending of inheritance. Okay, so he noticed all of that. What he did know was that each parent got to contribute to the offspring. Okay, so he did notice that. So in terms of what each parent's genetic contribution was to the offspring, each parent gets to give half of their DNA to the offspring. Therefore, each parent must have given one copy of their alleles. So everybody has two alleles for each trait. So because your parents each gave you one copy of an allele, so mom gave you one, plus dad gave you one, you have two alleles. So for every trait that you can think of, every characteristic that you have two alleles for that trait or that gene. Now, Gregor Mendel was actually quite lucky, and looks like this little, let's see if I can, yeah, I can't move the face recording, but what's covered up here is he chose simple test subjects. So Mendel chose simple test subjects. And in other words, he was able to use plants because they were easy to cultivate, they were easy to use, and simple to observe. And he happened to choose the most easy to observe traits that only had two forms. Okay, now that's not always going to be the case, and you'll see that. What you did in middle school were all of these easy to observe traits with two forms, dominant and recessive. But we're going to very quickly get out of the realm of that and go on to more complicated forms of inheritance. And if Gregor Mendel had actually chosen one of those forms of inheritance, he never would have gotten off the ground. So he got really lucky there. He also chose traits that obey very specific rules. Again, rules of dominant and recessive, right? The dominant is going to cover up the recessive. He also discovered something called the law of segregation. Law of segregation. Okay, and the law of segregation is this bullet right here. The law of segregation says that traits sort themselves between gametes. Okay, so in other words, your alleles get pulled into separate gametes, and he didn't realize that this was because of meiosis. We now know that meiosis is why, but the sorting of those traits is random. There's one other law that you'll see, and it's the law of independent assortment. Okay, he discovered that just because you inherit one dominant allele doesn't mean all of your alleles are dominant. It just means you inherited one dominant allele for that trait. There are other traits out there where you might have inherited a recessive. So that's what we mean by independent assortment. The inheritance of one trait doesn't necessarily impact the inheritance of a second trait. And he discovered ratios that are going to occur and reoccur. And when we get into the Punnett squares, we'll look at those ratios. All right, now associated with this is a little bit of terminology. It's not very difficult, but you have phenotype and genotype, and these words sound alike but mean two different things. And it's really important that you know the difference between phenotype and genotype. Phenotype is what you see when you look at an organism. So, for example, if somebody says, hey, what does your dog look like? And you say, well, my dog is kind of short, he has brown fur, it's long and curly, he has short ears and a long snout, whatever, something like that. All those things are phenotype descriptions. So what you see when you look at an organism is its phenotype. Genotype is what the genes or the two forms of the allele are. So, for example, big A, little a, little a, little a, big A, big A. And we'll get into how you assign these traits in just a little bit here. But these are what alleles do you possess. That's your genotype. Okay, so the genotype is what specific alleles do you possess. That's genotype. So oftentimes you'll be asked when you're doing Punnett squares to do a genotypic ratio or a phenotypic ratio, and often students will get confused between the two of those, and you want to be careful. If you're being asked for a phenotypic ratio, they're asking you, say, the difference between, or the ratio of purple flowers to white flowers, or tall plants to short plants, right? That's what you see when you look at the plant. You can see if it's tall or short, or purple or white. If you are asked for a genotypic ratio, then you want a ratio of these things, right, of different groups of alleles. Okay, so make sure that you understand the difference, because when you're trying to determine ratios in Punnett squares, that's going to be important. All of these things are using the laws of probability, and we'll practice with probability as we go throughout. All right. In this last little bit, what we want to look at is what's the relationship of a gene to its alleles. Okay, so let's use one of Gregor Mendel's examples. One of the alleles that Mendel used was plant height. Sorry, this is going to shake a little while I'm writing on the iPad. Okay, so plant height was one of the alleles. You had two forms of plant height. You had tall. Oops, come on. There we go, tall. And you had short. Right, tall and short. Those are the two alternate forms of the gene for plant height. So the characteristic is our gene, plant height, right? And then the two alleles are the two forms of that. Okay, so plant height is the gene, the two forms of that, tall and short, are the alleles that we are looking at. Alright, so how do you actually assign them the letters for the Punnett squares? And again, middle school you probably learned this, but you have to know which one is dominant, and the dominant you're always going to assign the uppercase letter, big T. Okay, so the tall here is the dominant trait, that's the dominant allele, and so we're going to call it big T. The short, being the recessive, is going to need to be labeled, and the rules for assigning the alleles to the trait are: the dominant always gets the uppercase, Okay, and the recessive is always going to get the lowercase. Okay, so the lowercase. And it needs to be the lowercase of the same letter. So the short is going to be little t. Okay, so we've covered what's a gene and its relationship to its alleles, and we've covered how do you assign alleles for the trait. We're going to do some phenotype genotype practice next, and you'll get a chance to practice with this.